of translation. So hopefully everybody's read Translation. There was a movie to watch last class while I was at jury duty. Good, that's over. So now we're going to see how transcripts are read and turned into proteins. One of the things that I asked was, here's a messenger RNA sequence. What's the protein that it encodes? So how, if you were a ribosome, which you have lots of, how would you read this transcript that's on the left? So eukaryotic mRNA sequence. What's the protein that's encoded by this transcript? CGG, AUC, GAU, etc. What do you do? What's the first step? This will be on the test.
Look for the first AUG.
Okay, so in a eukaryote, the ribosome binds at the 5' end and scans from the 5' end, it reads towards the 3' end looking for the first start codon. What's the start codon? AUG. Okay. So where's the first start codon here? AUG. You see down here that encodes the amino acid methionine, right? AUG, met, M, so the letter in the parentheses in all of these parts of the codon table is the single letter code for the amino acid. So most of, or all of the translation you should do, for me anyway, I'd prefer you to use the single letter code to tell me which amino acid a particular codon encodes. So AUG, we've got methionine. So that's how we know where the translation process starts. The first AUG you encounter as you read from left to right, starting at the 5' end of the transcript. That's eukaryotes. Prokaryotes have ribosomes that use the Shine-Dalgarno site that tells the ribosome where to bind and start translating. Now, the really important question is, how do you know where this translation process is going to stop? How long is the protein that's encoded here? How does the ribosome know when to stop reading the messenger RNA and stop producing a protein? It has to do with that magic word, stop. Stop codon. Okay, so how many stop codons are there? 3. Okay. They're all over here. I've written end instead of stop because end has 3 letters and I just like that better. But so there are 3 stop codons, UAA, UAG, and UGA. So where's this ribosome that's translating our messenger RNA going to stop? Who sees a stop codon? Over here. So they all start with U. UAA, UAG, UGA. When you spot one, tell me which line of the sequence it's on. Third. So there's a UAG there. Anybody find another one? Where are other stop codons in this sequence?
Right below it, there's a UAA.
So right below that one, okay, there's a UAA there.
No, it's the first UAA.
Oh, sorry, that one. So there are a lot of stop codons in this sequence. Which one is the ribosome going to use to stop translation? This is, at least 40% of you are going to try to do this on the exam. You're going to see a stop codon and you're going to say that's where the ribosome stops translation. It's not. Don't do that. So what do we have to do to know which stop codon the ribosome is going to use? We have to read codons, groups of 3 nucleotides, and figure out which of the stop codons that ribosome is going to read those 3 letters. That's what we call in frame. We read in groups of 3, and it's the first in frame stop codon that the ribosome encounters that will stop translation. So the best practice is just to underline groups of 3 successive nucleotides in the transcript. And then you see which of the stop codons is the first one that the ribosome encounters. In this case, it happens to be the first one that was identified. So that UAG is going to stop translation. Now to figure out what the protein sequence is, we just have to go and translate, using the codon table, all of those black underlined codons. We start with methionine. What's GCC? Right, we've got G and then CC down here. That's the next codon. Right. Alanine, A. CCC. P. Proline. AUC is isoleucine, I. GAG. Yep, glutamic acid, glutamine. GGG, that's the easy one to remember, if any of them. G, yeah. GGG encodes glycine. It's about the only thing that makes sense in this entire codon table. ACU. CAC is H. Okay. So what's the protein? It's some 9 amino acids, and then there's a stop codon. So this messenger RNA produces a short little protein, polypeptide, and then it stops. I've been using this example for term after term after term, and I still haven't gone back and fixed the original problem, which is I meant for this to actually spell something else. What should this read? It's not supposed to be map e-length. What was it trying to spell, geneticists? Map length. I was trying to be clever and come up with a protein that actually spelled something. Okay, so what mutation, this is something I love to ask. This is a really important concept to be able to perform. Imagine a mutation happens to this messenger RNA and the gene that encodes it. What sort of a mutation would cause this messenger RNA instead to make an amino acid sequence that spells map length? In other words, how could you change the AUC codon to, instead of encoding isoleucine, I, it encodes leucine, L. So here's that AUC. So what do you want to do? Okay, so there's an AUC. If we change that first A, if a mutation changed that first A in AUC to a C, then we would have a leucine. Then I actually would have done what I was trying to do and spell map length using a transcript. The point of all this is that mutations happen all the time to our genes. Sometimes those mutations happen in parts of the genes that encode the protein. So a mutation in the DNA sequence causes a change in the messenger RNA sequence that causes a change in the protein sequence, at least in this case. Where can mutations happen that don't change the protein sequence? What sort of nucleotide changes won't affect the sequence of the protein that's encoded by the transcript? In other words, when you look at the codon table, what sort of patterns emerge that are interesting or useful at least? How many different ways can you make a valine, V? down here in the lower right. There are 4 different codons that encode valine. So if you have a mutation, let's say we're talking about the GUU codon. What sort of mutations would change the DNA sequence and not change the amino acid sequence?
The last letter.
So if you change the last letter, if you had a mutation that changed that final U into a C, the DNA sequence changes, the amino acid sequence stays the same. Still have a valine. So that's one pattern that's interesting about codon tables, is that a lot of the amino acids are encoded by multiple codons. So there are lots of ways in which mutations can change the DNA sequence, but they don't actually change the sequence of the protein that they encode. What's the opposite of that? Which codons which amino acids are encoded by the fewest codons? Which ones are susceptible to mutation? Which codons, if you changed any letter to any other letter, would definitely change the amino acid they encode? What's another way for me to state that question? Which amino acids are encoded by a single codon? Tryptophan. So tryptophan, UGG, is only encoded by that one codon, UGG. If you change any of those letters, if mutation occurs in a tryptophan codon, I guarantee you it changes the amino acid that that codon encodes. The same is true for methionine. So those are the 2 codons where an amino acid is encoded by a single mRNA triplet. Then on the other hand, on the other end, we've got a few codons where 6 codons encode the same amino acid. So that's the span. We've got some amino acids that are encoded by a single codon, some amino acids are encoded by 6. So there's a huge variety in the codon table. There's a lot of redundancy. There are a lot of codons where multiple codons produce the same amino acid, and there's some where very few do. So one of the things I'll ask you to do at some point in the future is to design a mutation that causes a specific change in a protein sequence. And to be able to do that, really, you need to understand what we've just gone through, how a ribosome knows where to start translating a messenger RNA sequence, how it knows where to stop, and then how to use the codon table to translate the 3-letter codons into amino acid sequences. We've already been talking about this, how mutations impact proteins. If you get a DNA sequence change, it depends on where that DNA sequence change, that mutation occurs. What's the general trend in the codon table? Is it better if you had a chance to pick where mutations in your genes happen? Would you rather them happen at the first or the second or the third position in a codon? That is, where do all of these codons, where multiple codons encode the same amino acid, which position is it you could change the nucleotide and it encodes the same amino acid? It's generally the last letter, right? So it's generally the third position. So that's why all of these are boxed in this table. Every time that a single box has the same 4 amino acids, those are boxes, sets of 4 codons. And I'll stop there, but you could keep circling, right? Most of the codons are what we call degenerate at the third position. It doesn't matter what nucleotides at the third position, they all encode the same amino acid. And that means that when you look at human genes and genes from other species, most of the time you see mutations happening at the third position. Because it doesn't change the amino acid that's encoded. So evolution and natural selection tolerates mutations every third nucleotide. in a protein coding sequence, because those mutations don't do anything. They don't change the protein that's encoded by the gene. On the other hand, can anybody find a situation where you could change the first letter in a codon and keep the same amino acid? I think there's only 2 places on the codon table where you can do this. So it's really rare to have a mutation in the first position of the codon that maintains the same amino acid. Well, anybody find it yet? Okay, so leucine, I'm home. Yeah, so if you have, you can switch back and forth between U's and C's in those 2 codons. So UUA or CUA, UUG or CUG both encode leucine. So that's one of the 2. So we're looking for patterns where the same amino acid is encoded in 2 different blocks in the codon table. The other one is arginine, right here. Where you could either have C's or A's at the start of those codons, and they still encode arginine. So usually, mutations in the first position and the second position will change the amino acid. Most of the time, a mutation in the third position of a codon won't change the amino acid. Sometimes it will, a lot of the time it won't. So if you had the chance, hopefully you would pick not having mutations in the first or the second positions of your codons. Third, usually okay. Because we've all got tablets, I want to be able to involve you in actually mutating, analyzing, interpreting how mutations affect protein coding genes. So that's kind of the exercise for today. It all starts with this particular gene sequence. I went onto an internet database and downloaded this CDS, which stands for coding DNA sequence, of a gene called NUO1. I just chose one at random. So here you see the DNA sequence of the coding DNA sequence. This is everything that was transcribed into messenger RNA minus, because this has already been spliced, this does not contain any intron sequence. So this is showing you a messenger RNA sequence, except it's used T's instead of U's. Hopefully that doesn't freak anybody out. It's a messenger RNA sequence, essentially. And what's colored with the background shading are the different exons. So yellow here is exon 1, and then we've got exon 2 next to it in a different color, exon 3, exon 4, exon 5. So this gene has 6 exons. What are the letters that don't have color behind them at the start and at the end? There's 4 nucleotides at the start of this sequence. They're in lowercase that also, at the same time, don't have a background color. What does that mean? Say that again? What parts of messenger RNAs are not part of an exon? This happens to be what's called the untranslated region. So at this end of the messenger RNA is the 5' untranslated region, or UTR, untranslated region. Down here at the bottom, which I can't draw on, is the 3' untranslated region, the 3' end of the transcript. What do you think it means that they're untranslated? It's what it sounds like. Where's the start codon here? Be a eukaryotic ribosome. Where's the start codon? We're about to translate this messenger RNA. What is the start codon? What 3 nucleotides? No? Okay, go back to the codon table and tell me what the start codon is, methionine. AUG, or ATG in this case. So where do we see the first ATG show up? So it's the first, right, it's the first set of capital letters. So there's 4 nucleotides, and then there's an ATG or AUG. So what it means to be a 5' untranslated region is that's part of the messenger RNA molecule. It's just that the ribosome reads across that part of the transcript until it finds the first AUG, the start codon. Hence, those 3 nucleotides are not translated. The ribosome doesn't read them and turn them into amino acids. It's an untranslated region. What should that tell us about the 3' untranslated region down here at the bottom? The white background, lowercase letters. What's just before the 3' untranslated region begins? TGA, what's that? It's the stop codon, right? So all of the colored, the background color tells us, hey look, the first part is the start codon, methionine, and the last codon is the stop codon, asterisk. All of the messenger RNA transcript that's past the stop codon doesn't get translated, it's untranslated. Ribosome stops translating at the stop codon. So all we really care about is the sequence that's highlighted, the exons. That's the part between the start codon and the stop codon that the ribosome is going to translate. So if I was really mean, although it's Friday, so I'm feeling pretty nice today, I'd ask you to translate all of this and tell me what's the amino acid sequence of this protein. But, you know, we've got tablets, so don't do it by hand. Here's a useful website to use to do translations of messenger RNA sequences for you instead of using the codon table. So if you want to know the amino acid encoded by that CDS, coding DNA sequence, you go to this URL. There are lots of these web tools that are available. This is just one I happen to pick. It's not the best one necessarily. You go to this website, and right here, you paste in, oh my goodness, I'm using the lightsaber pointer. You paste in the CDS sequence there. The force is strong with me. Yeah, damn straight. And then all you have to do is down here at the bottom, just pick submit. So you paste in the CDS sequence, and then this computer program, the website, of course, just has the codon table built into it. It reads the CDS in groups of 3 nucleotides, and then tells you what they translate to. And so I paste in, I'm just walking you through this because I'm going to ask you over the weekend to do this yourself for your gene. So you'll have this recorded, it'll be on the YouTube movie, but I wanted to show you in class in case anybody had any questions. It's just this easy. Take a CDS, a gene sequence, paste it in, hit submit, and then in a slide I'll show you what it looks like when we're done. So that's the program Transeq. It's a website, you can paste in a gene sequence and it tells you what protein sequence a ribosome would produce. What I want to know, one of the things I want to know from you over the weekend as you work on this exercise is how many amino acids long is the protein that you're working with. So there's an easy way to do that. You need to be able to take a protein sequence like this. So just a random protein sequence. I can count that pretty quickly. That's 9 amino acids in a stop codon. But the genes that you'll work with might be hundreds or longer of amino acids. So I don't want you to do it by hand. So what would you do to figure out how long an amino acid sequence is without doing it by hand? Right, so that's normally what I would do is I would paste it into Word and then use the character count tool to tell me how many characters are in the sequence, and then I'd know how many amino acids it was. If you don't have access to a program that does that, then you could just go to the website that does exactly the same thing. So you paste the protein sequence into carecounter.com and it tells you how many characters, single letters, single amino acids, are in the sequence. So here finally is the amino acid sequence of the translated NUO1 gene. And what you don't want to do, just for information, when you use Transeq to produce the translation of a messenger RNA, it will always end with an asterisk. That's representing the stop codon. You don't want to include that in your count of how many amino acids are in your protein, because that's not an amino acid. So when you copy and paste something out of Transeq, don't also take the asterisk with it. So it turns out this has 479 amino acids in it. Any questions, concerns, comments? What we're going to be doing is taking CDS sequences, translating them, and asking how long they are. Which is sort of boring at first, but I want you to act like Mother Nature. I want you to produce mutations to this gene. And I want you to see what affects those mutations that you define, that you create. How do those affect the amino acid sequence of the protein? So here's how this works. I'm going to go through the description of the process, then we'll break up into groups after you do this part. So at the beginning of class, I gave everybody a Google Doc that has 2 copies, one on top of the other, of the NUO1 CDS sequence. So the same one I used in these examples. The top one I'd like you to leave alone. Don't change it. Just leave it there for your reference. The unmutated copy of the gene. And then to the second copy below it, same sequence I've given you in this Google Doc, I want you to choose how to mutate that sequence. It's up to you. I want you to make a single mutation, and these are the rules. If your last name starts with A through L, then you're going to insert new nucleotides. You're going to come up with A's, T's, G's, and C's of some length and stick those into one spot in the sequence. If your last name starts with M through Z, you do the opposite. You pick some part of the gene to delete. You highlight a few letters and delete them. So if you're inserting, either write out 1 to 6 nucleotides, A's, T's, G's, or C's, that you want to stick in. Just 1 through 6. So don't make huge insertions or deletions. Let's keep it small at first. You'll insert 1, 2, 3, 4, 5, or 6. Or if you're deleting, choose 1 to 6 nucleotides in a row from the existing gene sequence to delete. And then what happens is, if you're inserting, you're going to be in a group. We're going to break up into 6 groups in just a minute. We're going to have big groups. If you're inserting, you're going to be in a group that starts with I. If you're deleting, you're going to be in a group that starts with D. And then if you are inserting or deleting 1 or 4 nucleotides, you're going to be in group 1. 2 or 5 nucleotides, you're in group 2. And then 3 or 6 nucleotides, you'd be in group 2. Now is the good time to open that Google Doc. Scroll down to the second sequence. Make your mutations. Raise your hand if you've got any questions. When you're done making the mutations by yourself, go on to Socrative, and you can finally answer the questions. I know some of you have been in Socrative for a while trying to figure out what the questions meant. Now you know. Tell me what your group is, depending on whether or not you're inserting or deleting and how many nucleotides. And then predict these 2 questions. Okay, we'll come back together in about 5 to 7 minutes and discuss what you've done so far. So mutate freely. It's more important you know what you're doing, according to that table. Are you inserting your nucleotide? No, use the group that you should be. Okay. When you get together with your group. Insert. Okay, the good news is, if there's any good news, is that you don't really have to have done this right now. The important point was to try to make the mutation, but more importantly, to make these predictions. So once you've decided if you're inserting or deleting, well, that's based on your last name. Once you've decided if you're going to do 1, 2, 3, 4, 5, or 6, make these predictions. Now what we'd like to do, so if you're not done yet, that's fine. You can do the rest of this process over the weekend. Let's get into groups, though. So here's a map of the classroom. So we're looking at the front of the class is at the top here, up by me, front of the room. So I want everybody who is inserting, if you chose to insert 1, if you're in group I1, 1 or 4 nucleotides come up in this area. Behind them would be insertions of 2 or 5. Behind them at the back, insertions of 3 or 6, and then deletions. Okay? Couple minutes, then we come back together and we're going to find out what everybody predicted. Okay. So if it's an insertion in between the start and the stop codon, you'll increase the length.
But you also have the potential of prematurely stopping it if you.
Or you could prematurely stop, so it could decrease the length. Will it leave it the same length?
That's if you happen to put it before a start or after a stop.
Okay, so we're making good predictions now about how different types of mutations might affect translation. What about in the back? 3 or 6 nucleotides. Do you do something special that these 2 groups don't? Who's reporting out for your group in the back? Oh, I get it. Okay, so I'll put a no in for now. So, but you said it could either increase or keep it and not keep it the same length?
It depends.
The punchline to one of my favorite jokes. It's a bad joke, so I'm not going to tell it to you. But okay, what about deletions? Are we going to see a different pattern when we talk about insertions versus deletions? Is anybody in this first group for deleting 1 or 4 nucleotides? Any of you guys? You're all 2 and 5? Okay, so let's work as a committee of the whole. If you delete 1 nucleotide or 4 nucleotides from a coding sequence, what effect will that have on the protein length? Should it decrease it? Okay. What did you guys and gals say about deleting 2 or 5 nucleotides? Depends.
Well, I was going to say it's probably not that much different than what they were saying because when you delete, 2 or 5, it's an odd number, so it's going to change all of the reading frames. So the stop codon could be farther or closer.
Okay, so it could either increase or decrease, may or may not keep it the same length. Okay. So then group in the back. Delete 3 or 6 nucleotides. What happens?
So we agreed on that it would be a shorter sequence.
Okay. So if you make a deletion of 3 or 6 nucleotides, it should shorten the sequence. Does it have an effect on the protein sequence itself?
It can vary.
Okay.
But if it was a random deletion, you can have deleted one of the letters and it would become a top codon.
Right. So it still depends, in other words. So the point of doing this was to at least have everybody start thinking about the effects of different mutations, different types of mutations on the proteins they encode. Now, presumably all of you, 60 or so of you, have made totally different mutations to the same gene. Presumably, unless 2 of you collaborated and made the same change. It's unlikely that any 2 of you have the same mutation. So that's why over the weekend what I'd like you to do, and this won't take long, is to take the mutant version of NU01, where you inserted or deleted nucleotides, stick it into Transeq, get the amino acid sequence out, and then figure out, just by character counting, is it longer or shorter than the original NU01 protein? So that'll help you assess that first prediction. Is it longer, the shorter, or is it the same length? And then just look at the actual amino acid sequences of the two and ask, do they differ, do they not? Did the protein sequence stay the same or did it change? And then on Monday, when we come back, we will be here Monday. When you come back Monday, we'll discuss what you've observed doing these random mutations. We'll see if what you observe matches the predictions we just put on the table. By the way, last thing, sorry, I know I'm just 3 seconds over now. I'm reverting what I said about doing a review session on Monday because I'd like to be able to have this discussion, and this will be material that will be on the exam. So I would like to switch back to a Wednesday review session because there will not be a strike, as far as I know. So we should be here Monday, Wednesday, Friday. So Monday we'll have class, Wednesday will be a review session, Friday we'll have the exam.